and thank you, Marjorie, for coming. So we wanted to talk, and I will start off by saying, Master Gardeners will have like the second half of the meeting will be about what's going on and all that, just so that we can accommodate for Marjorie's time. Um, and so we've been talking about Laurel Wilt, and in the last meeting, I know you brought it up with Kathleen and Ryan. So I wanted to have the expert come in and talk to us about it so we can get a little bit more information. So thank you for coming, and we're looking forward to, or kind of looking forward to hearing what you have to say.
Thank you, Hillary. It's always a little difficult being the disease speaker. Does anyone really want to hear about it? And I would say I'm a pretty unwilling expert on this one. For months and even years, I've been telling people to quit asking me about Laurel Wilt because it was so far away. It wasn't going to be our problem for a long time. And then lo and behold, this summer it landed in my lap and I can't ignore it anymore. So any expertise I have has been gathered through the people to our south who've had actual experience with it for some time. And I do think it's important for us to all understand it. We're learning from it every day. We're coming to the hiatus period where in the winter it won't be making any more strides. But then next spring we're going to start seeing more of it, and it may be in your neighborhood. So let's talk about it. And then you're certainly going to be welcome to ask questions at the end. And if you can't wait, you can ask a question in the middle of it, all right? Let's see if I can make the slides advance. We didn't test that part. I found something to do it. Okay. So Laurel Wilt has a name that's going to confuse everybody and get them hysterical. It has nothing to do with mountain laurels. So you need to remember that it says laurel based on the fact that the plants that are affected are in the Lauraceae, which is the laurel family, but that doesn't include mountain laurel. They're ericaceous plants with rhododendrons. So pay attention to the actual host range and don't panic about mountain laurel. That's a really key thing to know. This is one of the, unfortunately, many invasive exotic diseases. It wasn't in North America until this century. The fungus is now called Harringtonia loricola, which is good because it used to be called Rafflesia, which is really hard to say. And I knew Tom Harrington, so I like this name better. The vector of it is really important because it can't get around without its vector. It does not have any wings. So the vector is a teensy tiny ambrosia beetle called the red bay ambrosia beetle, Xyloborus glabratus. And the detail on that is that we strongly suspect that other beetles might be spreading it as well. But in terms of our site here on Long Island that has been ever widening, we have found the red bay ambrosia beetle here. So we know that when this disease arrived here, it arrived along with the beetle. The hosts I mentioned are members of the Lauraceae, and the beginning of the infestation in North America, as far as we know, was 2002. That was when we figured out that it was here at any rate. We have had people commenting about things on sassafras for at least the past 5 years. Sometimes it's because they've heard of Laurel Wilt, so they're wondering if they have that. But in general, there are various reports over a wide area on all of Suffolk County where people will say they've seen wilt, they've seen dead leaves, like this tree, which was photographed in Oyster Bay in 2019. And another site in Oyster Bay has also been noted with some wilting trees. Mastic, which is down on the South Shore, has had some older trees that started dying right after winter a few years ago. And then even out in Orient, which is the furthermost tip of the North Fork of Long Island, there were groves of dead trees. And there we saw cankers on them. You can perhaps see in that picture, there are actual specific dead sections where the cambium has been killed on the trees when you investigate them. And as far as I could tell in that instance, it was from the canker fungi. And I think the other trees that have died up until now have largely been dying because of a combination of drought with perhaps armillaria, which follows drought often, the armillaria root rot, or some of these canker-causing organisms. We see Botryopsferia often, for example. And no one is really an expert in sassafras diseases who has done anything other than study this disease. because it's not generally used as an ornamental. It just so happens that it's a tree that we often have on our properties and we love it. So it's a tree of emotional significance, if nothing else. And we would certainly prefer to have it alive because it has those fetching little leaf patterns. And I'm not the only kid who had their daddy explain to them all the different kinds of mitten shapes that you could get out of a sassafras. Um, our instance that I'm reporting on today is associated with a call that we got from Northport. So, following upon a number of comments that came from hither and yon that said, well, this sassafras was bad and that sassafras was bad, where nobody ever sent in a sample. This Northport case was reported to Cornell Cooperative Extension of Suffolk County here in Riverhead, and the owner of the property was a organized and determined person, and she was interested in having a visit, and so we decided to go visit this property and see what was going on, particularly since she said that she had had a sassafras die last year, and now another sassafras was wilting on her property. That was intriguing. So we set off at the end of June to visit her property. Northport is towards the north of Long Island and sort of in the middle, okay, to give you a vague idea. It's east of Huntington. And this is Mina Becerra, who is our nursery and landscape extension specialist, and she's holding a wilted branch. One of the things we learned very quickly is it's quite hard to photograph wilt, especially when you're dealing with a forest tree. There's always something behind it that keeps you from seeing it clearly. But you, as master gardeners, all know what wilt looks like, so that's not really a problem for you. But Mina and I both visited this area and did what we could to try to photograph the way the trees looked before we started carving on them. And this one tree was, oh, probably about 4 inches diameter at breast height and definitely in a state of wilt, but still green. And that was part of what helped us make a diagnosis here that led us to the conclusion that we had Laurel wilt on the island. And here's our determined homeowner who turned out to be quite helpful. She even bounced into the house at one point and got her own chainsaw and came out and helped us cut her tree down, which was really helpful. And not everyone will do that for you. And she was really interested in finding out what was wrong and actually belongs to a native tree group in that general Huntington area where everyone's quite interested in native plants, and she couldn't wait to report things to the other people on her listserv there. We did what we could to document symptoms, which was useful because this turned out to be a really prime example of what we were going to need to be looking for later on. If you take a chisel and cut into the side of the tree, just wherever it's comfortable to cut, and you see something like this, you very likely are looking at laurel wilt. There's a blackening that's quite similar to what you see with verticillium wilt, if any of you have run into that before. Verticillium wilt would be more likely to be seen on a maple, particularly a Japanese maple or a Norway maple. If it's a sassafras, although it could be verticillium wilt, there's every chance that it's laurel wilt today. So this is a really nice sampling technique, and I'll show you another image of that in just a second. If you look at that same kind of discoloration in the xylem of the tree, by cutting across it, you're going to see little dots because it's not always continuous. It can be, but you should see black dots in cross section fairly near the outside and sometimes an actual black line just underneath the bark in the youngest of the wood. And then here's that same sort of continuous black sheath look as seen when chiseling into the side of the tree. And it's actually a very safe way to take a sample. You don't have to deal with saws. You can just hold a chisel in one hand and hit the back of it with a hammer and keep yourself pretty safe, and yet slice off individual pieces of that discolored sapwood. And that becomes a really good sample for us in the lab because that way we don't have to go get to saws and so forth to get at the part of a sample that we want. A log is not as useful to us. So if you leave the tree standing and take some vertical slices that encompass that darkly discolored area, that's perfect for anyone in the lab, whether we're talking the lab in Ithaca that's Karen Snovercliff's and Sandra Jensen's lab, or the one down here that I operate on Long Island. We did try considering all sorts of possibilities when we visited this Northport location. We were certainly thinking of verticillium wilt. I did everything I could to tell this homeowner about verticillium wilt and say I really thought that's probably what it was. And we do see it from time to time in different plants each time. There's a bottle brush buckeye wilting from it up in that picture. I think that was actually at Mohonk. Um, in addition to the possibility of verticillium wilt, there was a talk at the Long Island Horticulture Conference this last spring, in which Olivia Bigham, who's a PhD student at Purdue, gave a presentation about a new species related to the one that causes Dutch elm disease affecting sassafras. And she's been studying this for a couple of years, coming at it from really more of an interest in the vectors, the little ambrosia beetles that spread it. And so she gave a really nice presentation, and I was perfectly open to the chance that maybe this wasn't verticillium wilt. Maybe it was telling us of movement of her special new Ophiostoma on sassafras. But it turned out that those were both pipe dreams, because when we cultured, which we did on potato dextrose agar, we got these really funny looking colonies. They were white colonies, but they really looked very ragged on the outer edge, and they all looked very wet, as if they were contaminated with bacteria from the word go. And yet, when we looked into it, turned out that all that wetness was just an accumulation of the conidia, the spores of the fungus, which conceivably would be produced in the same way inside the vessels of the tree and would be what would be sucked up the tree to allow effects up high, even if the ambrosia beetle might have fed down low. So here's my classic bad hair day picture. This is the way an individual colony can look. Not all that different from other fungi, except in how ragged it is at the edges and how ropey and wet it is in its texture, which I hope is the only way it can look, because I've been using that as my designation. And we took the cultures that we collected from that first tree and sent 4 of them down to Florida. Romina Gazi-Saragina is someone who runs a diagnostic lab there and also does research on the avocado disease caused by this same fungus. It's also laurel wilt, but avocado is in the laurel family and the Lauraceae. So they've got an actual agricultural problem on their hands. We don't really have an agricultural problem now that they don't like us gathering sassafras roots anymore for tea. We just have a native plant problem, I guess, is probably the most important concept here. And Romina was able to identify using a series of tests. She did a LAMP assay, and then she did a PCR, and then she did some sequencing, and she came up with Harringtonia loricola, and it looked right to her as soon as she opened the package on top of that. There were subsequently several findings that went beyond Northport. They're all in the general area of Northport, though if you know Long Island, this will have more meaning to you. But St. James and Dix Hills and Hauppauge and Greenlawn, Fort Salonga, and Medford are all places from which we've confirmed the disease at this point. And Newsday got excited. DEC put out a press release, and Newsday bid on it. And they did terrific reporting on it. I was amazed. First of all, they were really quick. I think they called the same day the DEC press release went out. And then the article was there the next day. So they don't let any moss grow on them. And they showed the symptoms, which was very helpful, and also showed this sort of ring of trouble where the laurel wilt had been found. And they're showing it as sort of a Bermuda Triangle here with Northport and Greenlawn and St. James, the sides of the triangle. Since then, it's gone further south than this and a little bit further east. But we have not yet confirmed the disease in Nassau County. So if any of you have friends in Nassau County, you might get them primed to be the first in their county to find the disease. Since we can't fight it very well, being the first to find it is some sort of a scientific achievement, at least. Here's a map that Kelsey McCullough at DEC did to show the townships where the disease was occurring. Again, you've got that cluster of red stars, which tells you where samples that the DEC collected with their foresters became positive in culture. And then they were also making observations. So they wouldn't bring every single tree that had a symptom. They would make note of symptomatic trees, which are um, included here. And then they would also occasionally sample and we would get a negative. So all those things are included. And you can see that the disease, um, for the most part, has been most active on the North Shore of Long Island. So it will probably be a race between Connecticut and Westchester who's going to see it next. We'll see. It has to do with um, the flight of a very tiny beetle. I think it would probably be able to catch a wind across the sound. But there's also people, and people move things. So if you do see a new case, I would personally like to hear about it. But also you can report it by way of IMAP invasives. That is what DEC is monitoring. So they will contact you if you're in an area that has not yet been reported. They're particularly looking for new counties. And cooperative extension is linked into all this. So certainly if you tell Hillary, she's going to make sure the right people know about it. And we do have a lot of concern about the lower Hudson Valley. And I think all of our efforts to try to get people to manage the disease right now are directed at keeping it on Long Island and not letting it get into the Hudson Valley. I don't know if the bridges are enough of a barrier, but we would prefer that it not move. One of the things I've done here is to do something which is called trying to prove Koch's postulates. This is a terminology that is something that plant pathologists are familiar with because when you take a fungus out of a tree that has a symptom, like wilting in this case, if it's something new, you really do need to prove that the fungus you took out is the one that caused the wilting. You can't just assume it. There are a lot of fungi inside trees. So I was able to get sassafras and spicebush from a local nursery that were healthy and brought them in the summer and inoculated them low on the trunks and left 5 other trees wounded, but not inoculated for contrast, and just set about watching them to see what would happen. When we made the wounds, we made about a centimeter long wound with a razor blade and then put a little blob of culture on there that had spores on it and then wrapped it with parafilm to hold the area so that it would retain some moisture. And we did our inoculations on August 22nd. Within 2 weeks, we saw some yellowing on one of the sassafras, but it didn't progress to wilting initially, so we weren't sure what we were seeing there at first. The spice bush, however, were really dramatic after only 2 weeks. So we had very clear wilting, yellowing, necrosis, leaf roll on those plants that we had inoculated with the fungus. It was very, very easy to kill. And this might have to do with the fact that it's a tree that doesn't have one larger stem. It's often a multiple stemmed, more of a bushy type of a shape. I think that's part of the story here. At this point in time, no one has seen a spice bush with this disease in New York. So again, that would be a first. So if you would like to contribute to the scientific effort here, watch spice bush if you have access to those, and let us know if you see this wilting symptom, and check for the blackening that you were seeing in some of those first shots. When you have blackening of the vessels in a tree, it's a sign that it could be laurel wilt. And then it's our job in the laboratory to determine whether it's instead the other Ophiostoma from Indiana, or whether it's perhaps verticillium wilt or something like that. We did see leaf curling in the 5th spice bush that we had inoculated. It wasn't really doing any of the dieback, and it was mysterious looking. But over time, we realized that it wasn't actually about a fungus at all. This was where we had a little moth larva that was hiding in that leaf during the day. And it took us, I don't know, I saw it a few times and I finally opened it up to see if there was anything inside. And look who was inside. Really cute little fella. And this is an example of what happens when you have a native plant affected by an invasive disease from another continent somewhere. You have insects threatened as well as the plant itself. So the spicebush swallowtail likes plants in the Lawraceae, and it will be threatened by the presence of this disease. And it is a rather cute little larva, so it would be nice to be able to keep them around. Um, let's see. It was apparently just there to feed. We watched it eat all the leaves above this leaf where it was curled over. So it worked up this branch and just gradually cleaned it. So it was more effective than the disease in some ways, but it truly did leave the plant behind rather than taking the whole thing. Um, Later on, we started seeing symptoms in the sassafras. So the generalization would be that we saw symptoms in 2 weeks in the spice bush, and we saw symptoms in more like a month in the sassafras. And wilting was the main symptom. There was a little discoloration, but the wilt was really the most reliable kind of symptom. Things like this, where you see reddening, I'm still not sure whether that's a symptom of the invasion of the pathogen or whether it's a symptom of fall coming, because there are certainly a lot of red leaves out there now in the sassafras. But it's possible that that could be one of the symptoms of the pest. You We also saw an insect hanging out there too. It was really well camouflaged. So if any of you have blurry eyes, you're not even going to see this. But there's a gorgeous katydid sitting right there, and I walked past it 3 times before I saw it. It doesn't require this particular host, but it was enjoying it a lot for some reason. I think because it matched it well. Here's another one of the DEC images that maps out the populations on Long Island and into Westchester, showing you the the sassafras, and also the northern spice bush. And the spice bush is in the red and the sassafras in the purple. And you can see that where you live is very likely um, a congested amount of these hosts. So you're definitely likely to get the disease. How soon is a matter of people's behavior, particularly the behavior of people in that zone where we've seen the disease so far on Long Island. It would be possible to get a new introduction from the south, but I think the main question is whether people who go between the area of Northport and Westchester are ever so foolish as to put logs in the back of the pickup that are going to be used for firewood or something like that, and then dump them out in someone's backyard. That's the kind of thing that is not so much spreading the fungus as it's spreading the vector. The ambrosia beetle is carried in that fashion, and it sometimes takes its sweet time, but it burrows out of the wood and flies around and can go to nearby plants in the Lawraceae, which, as you know, have a nice aroma to them. So the ability of the insect to find its chosen host is actually quite good. And this is information that was put together from the iNaturalist reports of the trees. So that's what I have in terms of actual specifics, but I would suggest that you may have specific questions yourselves, and I can also add a few recent observations. Let's see, I'm trying to stop share, and all it's doing is giving me more ways to share. Are you seeing my screen now?
Yes. Well, we're seeing the PowerPoint.
Okay. So it doesn't too much matter. I'm going to try hitting that. Ooh. Let's see if
Actually, I think you want to stop sharing.
that's what I want to do.
I think I can do that for
Oh,
you.
okay. Please
Let's
do.
see,
Mm-hmm. I
unless
see
I'm wrong.
I see 3 more ways to share, but I don't see 3 ways to unshare for some reason. Oh, here, I did finally see one. I have things on top of things. There. That should have done it. Okay. Anyway, the end of my sentence is that I've been working with those same inoculated plants a bit in the past few days. And my job in terms of trying to prove that the fungus that we collected can cause the disease in these plants is not done until I re-isolate the fungus from the trees that I inoculated, and then we re-identify it as the same organism. And that can be done all the way to a sequence because we sequenced the first one. The interesting thing is that I was just letting it crank along for a while. And when I did my first isolations from spice bush, I actually was not able to pull out the fungus. So I just went back yesterday and did it again. And I used a different technique. We don't know all the details of how this fungus operates. So rather than using a Clorox bleach surface sterilant to clean off the competing organisms, what I did yesterday was to dip a section of stem into ethyl alcohol and then flame it, which kills things off on the surface, but doesn't actually heat up the inside of the tissue very much. And you don't have the potential for bleach to gas out what it is you're trying to grow. So it's too soon to tell, but I'm hoping that that method will help. But it is interesting because it does point out that even though we get a hint of where to look by where the tissue is very black, where the vessels of the tree are black in the sapwood, um, we're not actually seeing the fungus when we see that blackening. You're seeing the tree's reaction. So one of the things that I'm hoping to understand more over the next year is that if you, let's say, have an ambrosia beetle attack the tree 1 foot up from the base, is the fungus itself making it all the way to the top of the tree, or is it producing a toxin that triggers the tree's response, and it's the tree's response to the invasion that kills it, because trees do shut down their normal movement to try to stop the parasite from coming in, and they could, in essence, choke themselves off. So we're doing what we can to learn where is the most reliable place to find the fungus to confirm the identity of the pathogen. And I'm learning a lot from having inoculated these trees. I think the sassafras will probably still have the fungus alive in them because they have a lot more green tissue on them since they died more slowly or started to die more slowly. But that will be a job for tomorrow, and we'll see what happens with that. And also anything that I can't identify here by culturing, I can send up to Karen Snovercliff because she has the ability to do a molecular DNA reading technique called PCR, and she can look for evidence of the fungus, but it doesn't have to be alive. So dead fungus material in the tree would show up with her technique. So she may get positives where at the moment I've got negatives. So let's see, one thing to mention, because you're going to ask, someone's going to say, can this be controlled? Um, there is one thing that I know of that's labeled for control of Laurel wilt, and it's sort of accidental. I'm sure they had in mind that it be used for control of Laurel wilt way to our south. The disease was originally on the border of North Carolina and Virginia this season, and that's why I'm talking about there being a huge leap northward when we saw it on Long Island. The product that has a labeling for Laurel wilt control is one that's applied, it's an injection treatment that's done with microinjection called Phosphojet from the Arbojet company. And so if someone has a tree, it would need to be 5 inches in diameter. They don't do this kind of thing to little skinny trees. But if you had a really large sassafras, and some of them do grow quite large, that treatment would be a possibility for you. So that's already on the books as labeled for use in New York and other states. So there's an option. That's using chemistry that we already use for controlling Phytophthora on beech trees. And we also use that technology to control beech leaf disease to some degree. And we don't understand how effective it is, but it's definitely better than zero. And it seems to assist in cases where broad form is being used to control the problem, and to assist in cases where azadirachtin is being used to control the problem too. So, does anybody have questions? You've been quiet, so.
And anyone, feel free to unmute and ask a question if you have anything. And I'll take this moment to also remind everyone that tonight I have Margery again talking about beech leaf disease. So if anyone is looking for something to do tonight from 6 to 7, we have a webinar. So feel free to go to the website and sign up.
Are you
Happy to have
seeing
you.
this, are you seeing this in trees other than sassafras and spicebush?
We are not. In the rest of the world, they occur in other trees, but our ecosystem in New York really doesn't seem to have known hosts other than spicebush and sassafras, and we haven't even seen it in the spicebush yet. And I think that's because it's all about the vector. And I think the beetle is much more attracted to a standard tree trunk. It can't really tell one bush from the next as well. So I think it's more likely to go after sassafras. So probably we'll only see spicebush affected in areas where a lot of sassafras have been affected before. And you can help me test that theory. We'll see. The two trees do seem to occur together in wild areas. So I can't imagine it will be too long before we find it there because it has been seen in the southern states to go to the spicebush. That wouldn't be shocking. It's just that we haven't seen it here yet. And then the red bay, which is in the name of the insect, refers to a wild laurel family plant that occurs from Florida up to the Carolinas. And it was decimated by this. It was sort of more of a, I shouldn't say trash tree because I don't think any trees are trash, but it was just something that lived along ditch banks and so forth, and people weren't paying much attention to it, but they noticed it when it started dying, and it died in great numbers. So thousands of trees have been killed by laurel wilt there. And that created instant concern because once it was identified, people figured out that avocados were also challenged, which they do care about a bit in Florida and other southern states.
Right here.
So, and if you grow bay leaves for your kitchen and you happen to take them out during the summer, those little trees could die. They would have to be attacked by the beetle again. But New York will probably be the first place where that happens naturally rather than in an experiment. But I think it's possible. I've been eating as many avocados as I can. I want to inoculate some avocados so I can say I have the first New York avocado with the disease. I think DEC is on to me, though. I don't think they're going to put it on the map, even if I do get it. So. But it is interesting. I still am worried that many people won't be able to recognize the spice bush because it's a relatively nondescript plant when it's not blooming. It's quite lovely when it's blooming. But if you are trying to make it out, all I can do is tell you that if you have ever grown an avocado from seed, and I'd like a show of hands of the people who are on the screen, whether you've grown an avocado from seed, okay? There are a few. It looks a lot like avocado foliage, okay? A little bit narrower, but the texture is about the same as avocado, and the overall look of the young spice bush is very similar to a young avocado. So, and now I can just imagine all your little master gardener minds thinking, oh, I'm going to eat avocados this winter, and I'm going to plant a lot of them out, and I'm going to get the first avocado in New York. But you're welcome to compete with me. It's fine. Anybody else? It's an interesting disease, but we would have been happier without it. We
Does
don't-
does mulching the wood destroy the beetle or the fungus? And
Mhm.
do you then use that mulch?
That's a very good question. I meant to put that in the talk, so thank you so much for asking it. Mulching does destroy almost all the beetles, which is probably as good as we can get. I've seen people say to double mulch. I don't know whether people are willing to put mulch back in the chipper and do it all over again or not, but the more you create thin slices, the less likely a beetle's going to be able to feed until it matures and flies away. So if we find it now in this area around Northport where it's been discovered, which is an area, I guess it would probably be about 50 miles across at this point and across and down. We recommend to people that they chip the trees and they keep them on their property. So that's a double assurance because that way, if any beetles emerge, then presumably they'll be looking in that same exact place rather than we haven't given them a ride 100 miles away.
Right.
we figure that firewood is probably the reason why we have the problem in New York, that somebody had a second home in North Carolina or South Carolina or whatever and brought some logs up, and then some beetles flew out. And how long ago that happened, I don't know. There are a number of dead trees now. The counts you may have been able to see on that slide, they've looked at over 3,000 trees now of the DEC people who are surveying, and they found over 700 trees with death and wilt and so forth. That's not all confirmed as this disease, but a good part of it is, because it's something new and they've got an eye for it. But that seems to be the smartest thing now. If you could bury it or you could burn it, except that there usually are local ordinances that say you can't do the burning. But keeping them and using them yourselves, using that mulch is good. If you wanted to be ultra careful, you'd mulch and then cover with a tarp, but I think that's too much for most people. And you couldn't do an imperfect job. It would have to be perfect, because the beetles can crawl out if you leave them a little space. So I think it's fine just to do a thorough mulching job and leave the wood on your premises. And I hope none of you have a backyard that's 100% sassafras, because this could be ugly in that kind of situation. Because even if the trees aren't too large, if you have a lot of them, it would be really difficult. I've also noticed that the ones on Long Island have a tendency to be very tall trees. They've been seeking the light for years. And often the foliage that is showing the wilt is way up high, to the point where you need binoculars to see it. And then you can't really take a branch to use it as a sample, which is why this idea of taking a sample from the trunk is really helpful, because that's easily accessible to you. And you can get something that can be cultured and determined.
Okay.
Anybody else? Okay, we'll know more next year, but this is what we know right now. And I hope it will not go into other species. It seems to have a real affinity for this particular family of plants. But since we're not sure which beetle vectors it's going to start using in our part of the world, we don't know for sure whether it might not be carried to some other kind of tree or something along those lines. It's just an unknowable thing. We have to go through it and see what happens. It doesn't seem to be minding winter, but it also doesn't seem to be growing aggressively even in the fall, which is what I can assume from not being able to isolate it yesterday. So I think that we can be hopeful that the disease will not gallop along as much as it did in Florida and the Carolinas and Georgia and so forth. Okay, so I'll stop there and try to reset my head to think about beech trees. And if I start talking about sassafras tonight, just remind me. I did that one already.
Well, thank you for coming and talking to us about this.